I'm Ngoc Dang Do. I'm a physician scientist at the National Institutes of Health in the United States. And on today's webinar, I will be presenting information on CLN3 biomarker discovery and talking about biomarker in the context of translational research, particularly in human diseases and in CLN3. Initially, I like to start out with the definition of biomarkers that have been defined by a working group at the Food and Drug Administration and National Institute of Health in the US. Biomarkers are defined as measures that can be used to assess the normal cellular processes or disease processes and help in determining how the cells or organs respond to candidate therapies. It's also important to note what is not considered biomarkers. Measures of how an individual feel, function, or survive are not considered to be biomarkers. On the next slides here are some examples of categories of biomarkers on the left and their application. Biomarkers, as I alluded to, can be used to assess the susceptibility or risk, to diagnose a disease, help in monitoring how the disease progresses over time. Biomarkers also have application in predicting how the disease will manifest in the future or assessing how a potential treatment may have effect on the disease. On the right-hand side are different methods or types of biomarkers. There can be physiologic biomarkers. Some common examples are measurement of an individual's blood pressure, heart rate, and other measure of the heart or the brain activity. Again, examples of measurements that are not considered biomarkers, such as intelligence score, an individual ability to move or see, because these assess the individual functions and therefore they are not considered biomarkers. Biomarkers can also come from the area of histology that evaluate intracellular features. Some examples are immunocytochemistry of receptors, estrogen or progesterone, in the case for cancer application, measurement of intracellular accumulation, such as neurofibrillary tangles or cholesterol accumulation. In the area of radiology, there are also biomarkers being developed to look at the size of the organs of interest or look at the density of the organ of interest in certain bone diseases, or looking at the firmness of liver to assess disease processes. There's also biomarkers that are being assessed using molecular techniques. Here's term omic techniques such as proteomic, transcriptomic, metabolomic, or lipidomic. And here are some examples that have been used to assess inborn errors of metabolism. Application of biomarkers, these can be used to support applications for new drugs. And here are some data from a review that show number of new drugs applications that have been approved without biomarker here in the darker orange, and the number of applications that are approved with the use or supporting data of biomarkers. And this data has come from the European agency. So as you can see, the use of biomarkers has a significant impact on the number of new drugs that are being approved. Breaking this down into the different disease categories, One of the things to notice, applications that are approved with biomarkers in the lighter pink, salmon pink here, in most of the disease category, the use of biomarkers improve the chance or the success of having the intervention being approved. And some of the highest categories where biomarkers have a lot of success are in cancer therapy and autoimmune diseases. One of the most efficient or more optimal way of identifying biomarker or doing biomarker discovery is doing it as part of an observational or natural history study. Natural history studies are typically clinical trials that do not involve treatment. They are designed or pre-planned to assess or measure different parameters of the individuals and the disease, such as the genetic makeup, what else contribute to the disease progression or presentation, and other markers or measurements of the disease. There are different ways to conduct natural history study. We can do them retrospectively, meaning we look at data or samples that have already been collected. So we review charts or samples. The advantage of this is that the data are already available. Because they are available, we can identify certain lack of information or knowledge gaps. We don't need to spend a lot of time to acquire the data. However, since this were not done as a pre-plan or design study, these retrospective studies can have disadvantages in that the data or samples collected may not be uniform, and there might be missing data, or the data might not address the area of interest. Conversely, a prospective natural history study are pre-planned and involve future or ongoing collection of data and samples. These have the advantage that you can, again, make it more systematic so that the data and the samples collected can be more uniform. However, they require time as they are ongoing or future studies, and the knowledge gaps may not be as readily apparent. In somewhat of a parallel or similar concept, that you can look at one time point or cross-sectional natural history study. These are typically shorter in terms of the study duration. They may have application for therapies with immediate benefits, but they might not give the full picture of the disease course. Conversely, longitudinal natural history will collect samples over time, so they may give more information on how the disease progressed, but they are time and resource intensive. This slide here gives some examples of observational or natural history study that are ongoing or have been done worldwide in the field of neuronal ceroid lipofuscinosis, in which CLN3 belongs. So there are studies in Italy, several in the US, and a long-standing natural history study of the NCLs in Germany. At the National Institutes of Health, we have been conducting a natural history study focusing on CLN3 since 2017. And we've recruited study participants worldwide. And this gives an indication of the number of participants from each areas of the world. In this natural history study, we do extensive clinical evaluations to look at multiple systems and functions so that we can correlate this with other measures. And by other measures, here are some examples. We do sedated procedures to look at certain brain imaging. We collect samples, including spinal fluid, blood, and urine. Since 2017, we've seen 47 individuals in person for the first visit, so baseline visit at 0. And then we follow these individuals, about 2/3 of them, on average on an annual basis, either in person or remotely. So in the rest of the slides, the format or the layout, I'll be talking about some examples of biomarker candidates in CLN3. And for each of them, I will try to highlight how they are being detected, how the samples are accessed, what is the link to the underlying CLN3 disease, how they can be applied, and what are some of the challenges or caveats. And these are biomarker candidates that arise from some of the natural history studies that I've mentioned previously. The first category of biomarkers that we'll be talking about are histologic biomarkers. One example of this is the fingerprint profile. These are seen by a powerful microscope, electron microscopy, and they can be seen in different tissues, including some blood tissues such as lymphocytes, but they can also be seen in other biopsy of other tissue types in individuals with CLN3. It is thought that the features that are seen, so here are what the accumulation in the cell at higher magnification. They have these patterns that look like fingerprints, and hence the name fingerprint profile. These accumulation are thought to be intercellular or lysosomal accumulation of waste products and materials in the cell. Fingerprint profile has been used to support the diagnosis of CLN3 when there's genetic or genomic tests showing only one variant. This can be helpful in confirming or supporting the diagnosis. As you might surmise, some of the challenges of using this is that it requires specialty expertise. So facilities that have access to electron microscopy, and also pathologists or individuals who can evaluate these slides and pick up these abnormalities. It is a qualitative measurement, and other NCL conditions and potentially other lysosomal conditions may have this pattern of cellular accumulation as well. So it is non-specific. Another histologic example is vacuolated lymphocytes. These are typically detected using light microscopy as a manual evaluation. And these can be seen on peripheral blood smear, so no specific or specialized tissue is required. And here are some picture examples from a Cooper et al. Publication of what these look like. In lymphocytes from healthy individual, you may see maybe one or no areas of clearings. So those are vacuolations in the lymphocytes. In individual with the classic presentation of CLN3, there are many more of these vacuolated bodies present within the cells. And then interestingly, in subtypes of CLN3, i.e. individual who might have a more protracted presentation, or individuals who have the vision involvement only and few or no other organ system being affected, there's fewer numbers of devacuation. And this graph at the bottom here recapitulate that idea, and that in these control healthy individuals, the number of cells with vacuolated lymphocytes are less compared to individuals with different presentation of CLN3. The potential application of this would be, again, to support the diagnosis of CLN3, and it perhaps or potentially can help in predicting or segregating the different types of CLN3. The challenge is, again, it is non-specific, so other disease condition may have this appearance as well, and it may be low throughput because, again, it requires a hematologist or a heme pathologist in order to evaluate these slides. From the same group, however, they have a follow-up work where they are implementing more machine learning types of analysis to see if this could be automated. And they suggest that there may be some success there. The next category of biomarker for which we'll talk about a few examples are radiologic biomarkers. So the first one is brain volumetric, so measurement of the size of the brain. So here's an example of a T1 weighted image of the brain showing the cerebrum and the cerebellum, the brainstem, and you can see the distinction between the gray matter and the white matter. The white matter consists of the neurons processes, and this is a little bit of a better definition between the gray and purple here and the white matter in green. So in terms of methods, T1 weighted imaging of the whole brain, which is typically present or accessible at any clinical hospital or center. The pathophysiology is that in CLN3, it's been shown that there's decrease in brain volume, which is thought to relate to the neurodegenerative processes or the loss of healthy neurons. Here's the natural history data from the German or Hamburg group showing that across the ages and within individuals with CLN3, as they grow older, i.e. as the disease progressed, there's a decrease in the volume of the gray matter. What's also helpful is that this decrease in brain volume correlate with the clinical score that depict more severe presentation of the disease. Application for brain volumetric, as you can surmise, is in monitoring of CLN3 disease. And if there's a candidate therapy, brain volumetric may be applicable in determining whether that therapy is efficacious, in that whether it slowed down or maybe even reverse this decrease in brain volumetric. Some of the challenges for this potential biomarker or method is that it required the individual to be cooperative because magnetic resonance imaging is highly sensitive. So any motion or movement can produce artifacts in the images. So for long imaging, sedation or anesthesia may be required. There's also some challenges with consistency. Different scanners or sequences used to obtain the measurements may yield slightly different images or ability to quantify these measures. The second radiologic candidate biomarker I'll be talking about are brain metabolic markers. So this is also done by magnetic resonance imaging, but instead of looking at the structure, this look at the level of certain chemicals in specific areas of the brain. So in this picture, this is in red square here, is depict what's called a voxel, or the specific defined area of the brain where chemicals such as N-acetyl aspartate, or NAA, which is released by healthy neurons, are measured. This peak here depicts creatine. It's also a compound that can be found in cells. It gives an indication of the cell's energy or metabolic processes. So the methods of accessibility is MR imaging of the whole brain. Again, the pathophysiology is as there's neurodegeneration or decrease in number of healthy neurons. Certain chemicals associated with healthy neurons, such as NAA, may be hypothesized to change or decrease. This can be used in the monitoring of disease, as we'll show in the next few slides, and also determining of the efficacy of potential candidate therapy. Similar to brain volumetric, the challenges deal with patient cooperation and consistency across scanners and methods. So here are some examples from the natural history study of what brain spectroscopy measurements would look like in individuals with CLN3. In these 3 different panels, we look at the levels of N-acetyl aspartate, that chemicals that released by healthy neurons. Similarly, glutamatergic is also chemicals associated with presence of healthy functioning neurons, as well as creatine. So again, across these ages of individual with CLN3, you see that there's a decrease in the level of each of these chemical markers of healthy neurons compared to a reference similar age cohort. The spectroscopy markers also correlate with the clinical measures of CLN3, represented here as the Unified Batten Disease Rating Scale measurement is another measurement of disease state, as well as a couple of measurements of neurodevelopmental status. So on this heat map here, the darker the color, whether it's blue or orange, or the higher the number here, depict stronger correlation between these spectroscopy markers with the clinical markers. So in the last group of biomarker candidates, I'll be talking about molecular markers. The first one of which is neurofilament light chain. It is the protein that is released upon pretty much any type of damage to the neurons or the processes of the neurons. Detection method, it is typically measured by an immunobased assay, and it can be measured from spinal fluid, plasma, or serum samples. The pathophysiology is that upon being damaged, the neurons are thought to release neurofilament light chain into cerebrospinal fluid, and a percentage of that gets excreted into blood or serum where it can be measured. Similarly to the brain biomarker candidate, the neurofilament light chain can be used to monitor disease and potentially to help determine efficacy of candidate therapy. The challenge here is that for any of these fluid biomarkers, we need to know a reference range. What are the typical levels in individuals, especially pediatric individuals, who do not have the condition, who do not have CLN3, so that we can compare and determine whether the level that is being measured in individuals with CLN3 are abnormal. And NEFL can be less sensitive in plasma or serum because, again, only a portion of the CSF neurofilament light chain is excreted into those compartments. Here is some data from the natural history study of individuals with CLN3 at NIH to show that in spinal fluid, indeed, the level of NEFL is much higher compared to individuals of similar age who do not have this neurodegenerative condition, whether they are healthy or they have other neurologic but non-neurodegenerative condition. Again, the finding of neurofilament light chains and its increase also correlate with other clinical measure, including that disease state rating scale, UBDRS, as well as a measure of neurodevelopment and the MR spectroscopy markers that were shown earlier. So here, if the NEFL level correlate with these measures, then the measurements move away from this 0 point and actually do not cross the 0 point. So there's high correlation between NEFL and the CSF and these other measures of CLN3 disease severity. A more real world or real life application of neurofilament light chain is the assessment of its level in a phase 1/2 study looking at miglustat in individual with CLN3. So this data comes from a phase 1/2 study conducted by the Beyond Batten Disease Foundation and Theranexus, where 6 individuals, 17 years of age or older, were treated with miglustat, and their blood and spinal fluids were collected at the baseline and subsequently. This graph on the left here shows that on average, there is a decrease in the release of this NEFL, which is kind of a surrogate for a decrease in markers of neuronal damage. That decrease is seen more starkly in the CSF samples that we have fewer measurements for. The second example of molecular fluid biomarkers are glycerol phosphodiesters. These can be measured using mass spectrometry, using metabolomic assay. They can be measured from cerebrospinal fluid or plasma or serum samples as well. These are chemicals or compounds that are relatively more recently discovered to have a role in the pathophysiology of CLN3. The current working hypothesis is that there are defective clearance of these compounds from lysosomes, leading to further accumulation in the cells. They may have application in the diagnosis of CLN3, and there's current work going on to determine or assess whether they have application in the monitoring of CLN3. The challenges are similar to the NEFL challenges that I mentioned earlier. Here are some data or information on the application of GPDs. So this group have used cerebrospinal fluid from individual from the natural history study to show that there's increase in one subtype of GPDs called glycerol phosphoinositol in spinal fluid of individual with CLN3 compared to, again, samples from individual without CLN3. And the pathophysiology also extends to animal model of CLN3 disease. So here's an example of measurement of GPI in the mouse model of CLN3, where there's a significant elevation again of GPI in CLN3 mutant versus the wild type. This data have also been shown by another group looking at the pig model of CLN3, where over time, they also observe increased level of GPI in the CLN3 mutant pig versus the wild type. They also use blood samples from CLN3 study participant in the NIH Natural History Study and reported or observed that there's also increased GPI level in individual with CLN3 compared to family members who are carrier but are unaffected, or family members who are carriers who are not carriers and are not affected with CLN3. In modeling the results, there's a suggestion that measurement of GPI has a high degree of sensitivity or ability to help with diagnosing or identifying individual with CLN3. The methods of measuring GPDs or GPIs in the CSF, plasma or serum, may be a bit challenging because the level are typically low. So the group that had been working with at Stanford has been more recently looking at methods to increase the sensitivity of measuring these compounds in blood samples. So for example, using magnetic beads coupled to antibodies to pull down the lysosomes where these GPDs accumulated. So in summary, here are some characteristics of what biomarkers are. They are measures that can be used at supporting data or surrogates to look at the disease pathophysiology and to help in the diagnosing, monitoring, or assessing of the efficacy of candidate therapies. In CLN3 particular, we have reviewed some example, histologic example, radiologic and molecular examples of biomarkers that can be used to support the diagnosis, again, monitoring or assessing efficacy of CLN3 disease. Thank you. Best wishes.